Welcome to Temple University, and thank you again for all of your support for our research students. This year we had changes to our research program that were in part due to changes in the government. However, those changes benefited every research student at Temple University. What we formerly had done for our Valais students, we now did to a population of 74 students. Every student engaged in writing a research proposal. Every student worked on creating a milestone pathway and looking at their own research progress very reflectively. Every student did an elevator speech. Every student experienced preparation for graduate school. In just a moment or two, you will meet some of our students. I hope you were thrilled with the work that they did as we were. Thank you again for all of your efforts on their behalf. We really couldn't do this without your support.
My name is Sanjana. I'm a senior cellular and molecular neuroscience major here at Temple University. This summer I had the opportunity of working with Dr. Lu Chen at the Center for Substance Abuse Research at the Lewis Katz School of Medicine. My project this summer focused on diuresis, which is an excretion of water from the body without an alteration of electrolyte balance. So what I did was we studied the kappa opioid receptor, and we used kappa agonists, which are drugs that bind to the kappa opioid receptor. This summer I was working in the Center for Substance Abuse Research at the Lewis Katz School of Medicine under Dr. Lu Chen. I studied the kappa opioid receptor, which is what her lab is mainly focused on. My project specifically this summer was focused on diuresis of kappa phosphorylation deficient mice. Diuresis is essentially classified as an excretion of water from the body without an alteration of electrolyte balance. Kappa agonists, which are molecules that bind to the kappa receptor, are already known to cause diuresis by decreasing vasopressin, which decreases reabsorption of water into the body, so more water is excreted. Basically, what I was studying was the why behind this mechanism, or the how, and specifically if it included the process of phosphorylation of the receptor, which is a process that's really important for desensitization, internalization of the receptor, and then recruitment of a different signaling pathway as well. We basically had two mouse types, a mouse type with a bunch of mutations that made phosphorylation impossible from the kappa opioid receptor, and we had a mouse type that was considered wild type, which basically means they had no genetic mutation. And essentially we compared the diuresis produced by each when injected with these kappa agonists. Unfortunately, we did not find a link between phosphorylation and diuresis, but in the future I'm going to be studying this phosphorylation mechanism in relation to a pain response. So that's really cool. In the future, my plans, I'm going to continue research, maybe at the Lewis Katz School of Medicine, or maybe somewhere closer to home, which is Boston. And hopefully I'll do research for a couple years before considering professional school of some sort. And yeah, I thank all the funders for this opportunity to study here this summer. I had a wonderful time, and I learned so much. So yeah, thank you so much.
Hi, my name is Jenny. I'm a data science major here at Temple University. Over the summer, my research involved about generative AI and how they impact interdisciplinary learning. We started off back in spring 2025, and I decided to continue with over the summer. Over the summer, we conducted extensive literature review to get the idea of what there's out there and what we need to work on. What we learned over the summer was how bioinformatics is a very large field, and not a lot of people have put their hands on it, and how they are learning about it right now. So to overcome that situation, we made a chatbot which help students learning about bioinformatics and get them more involved and interested in the field. We also are planning to submit our paper for CHI, which is also known as the ACMW Conference for Computing on Human Factors Conference, which is like one of the top conference for involving bioinformatics or bioscientific measures. Over the summer, I also gained a lot of skills like literature review, working on front end and back end web development, literature review, research methodologies, data analysis. We also learned how to making a team more collaborative and involved within the research, and how to interact with chatbot to specifically dedicated towards the bioinformatic aspect of the whole research. I'm very thankful for this opportunity, and I would like to thank the funders who made this opportunity come true.
Hi, my name is Anae. I'm a genomic medicine senior at Temple University. This summer, I worked at the Fells Cancer Institute for Personalized Cancer and Medicine. This summer, I worked at the Fells Cancer Institute for Personalized Medicine under Dr. Grania, where we worked on understanding FAM122A in its recruitment to DNA damage sites. My project under Dr. Grania and Anita Kumar worked on understanding how cells react to damage. So every day our cells face stress that can break or mutate or damage DNA, and if not fixed, this can lead to diseases and mutations like cancer. So to prevent this damage, our cells have built-in checkpoints, kind of like traffic lights, that hopefully will block the DNA damage before it gets through and slips through the cracks. So scientists already know that FAM122A works with this process of the checkpoints, but how it gets to these sites of the DNA damage is still unknown. And also how, when it gets to these sites, how it interacts with the other proteins in this site is unknown. So in Dr. Grania's lab, we did a bit of work to understand this problem by utilizing different cell lines and making different vector viruses with different FAM122 knockouts. We used A, B, and C, and so we made these cell lines and we tested out different things and see how it reacted with the different proteins, if it survived. Now we're still in the makings and the beginning processes, but I was able to be a part of this beginning process, and it was a really, really cool experience for me, and also my first lab experience. This research is important because if we understand how FAM122A works, it will open up new ways to target cancer and make maybe new therapies that rely on understanding faulty DNA repair systems. In other words, if we know how the protein controls the traffic light system, then we may be able to design better therapies or treatments that slow down or stop cancer cells from multiplying. Through this project, I've learned a lot of new techniques. For one, I've learned how to work with CRISPR, which this is a tool that we can use to silence or turn off genes in the cell and see how the cell responds, or see how, in the absence of that gene, how the cell might be affected. Beyond this project, I see this experience as a really valuable time and has made me love research more and want to pursue research in my future academic career, whether in a more lab setting or in a more professional setting. It's definitely opened my eyes to seeing the scientific process and wanting to learn more and apply more. This research experience has had a big impact on me. It has helped me learn how to work independently in a research lab setting, but also collaborate as a team, as I was working under Dr. Grania's lab, which also had a postdoc, so we often came together to put all of our research together. So this experience has given me multiple experiences that I'll be able to put together and apply in my future career. Finally, I want to thank my mentors, Dr. Grania and Dr. Kumari, and then I also would like to thank the CST SURE program as well as its funders for allowing me to take the opportunity to learn more about science and working in the lab, and it's because of you guys that we're able to do funded programs like this over the summer. So I just wanted to say thank you, and this has definitely opened my eye to further research in my career. Thank you.
Hello, my name is Selena, and I am a chemistry major going to my sophomore year this semester. So my research title is the selective access to CF3 carbonyls or trifluoromethyl ketones on a variety of electron-deficient heterocycle molecules. We want to put fluorinated motifs onto a bunch of different types of heterocycle compounds because fluorines are used in a lot of medicinal chemistry. So about like 20% of all pharmaceutical products contain fluorines, and over 85% of drugs are have heterocycles. So we want to find a way to put fluorines onto heterocycles so that we can have a so that when people are synthesizing medicines, more they have more techniques and more pathways to access, I guess, this type of like fluorinated medicine. Specifically, we're focusing on trimethylcarbonyls and difluoromethylketones because there's not a lot of ways to access these two types of functional groups, and they're very important to serve as bioisosteres of carboxylic acids for pharmaceutical drugs. So we're trying to figure out how to insert these two types of functional groups onto a wide variety of heterocycle compounds. So for this research, I've learned a variety of techniques used in the organic chemistry lab, specifically techniques like synthesizing molecules or how to run a column chromatography. At first, it was a little hard to learn, but through running like over like 40 columns throughout the summer, I've got faster and I've got a lot cleaner products. I've also learned how to analyze NMR spectrums and to verify that I did indeed get the product that I want to get. For the future, we're thinking about publishing this as a paper. So for the future, we're thinking about making the CF2 ketones, and I think this summer has really impacted my professional development in terms of thinking what I want to do in the future. For now, I'm really interested in doing a PhD program and perhaps completing a PhD in chemistry specifically. Prior to this program, I didn't really know what chemistry PhDs actually entail. I actually didn't even know what a PhD program is. So this program has really helped me into giving me like more insights onto how PhDs work, especially seeing how my mentors and his colleagues are working in the lab. So I really want to thank my mentor, my PI, and everyone that has helped me throughout this process. Specifically, I also want to thank all of the funders for this program and to give me this opportunity to conduct research over the summer and hopefully throughout the semester and through all of my time at Temple.
Hello, my name is Kai. I am a junior mathematics and physics major here at Temple University. Over the summer, I worked with Dr. Darius Torchinsky in a laser lab, an optics lab. So what we did was we took a bunch of lasers and some mirrors, some lenses, and what was called a digital micromirror device, which is a tiny, about tic-tac sized mirror that's full of oscillating mirrors that go back and forth in binary. And when you point the laser at it with a pattern on the mirror, it creates all kinds of fun shapes that you can do a lot of stuff with. In particular, we were looking into quantum information and quantum computing, quantum key distribution, which is all very, very technical, but essentially what you can do with this laser is encode information and send it to whoever you'd like, who can interpret that laser back. So I learned a whole lot about lasers and light and things reflecting. I learned a lot about hands-on lab setups. I did so much reading on how to set up a lab. I did so much hands-on experience with adjusting mirrors, super, super tiny mirrors. And I did so much work with taking things apart and putting it back together. And I mostly was on my own here. So I learned a lot about how to do research as an individual. In the future, we're going to work on this and actually create a quantum informatics setup. So we're going to be able to encode information with that little tiny mirror, send it to some other place, probably within the lab, receive it, and decode the information that's on the laser. And with this laser, you can code about 30 lines of information at one time, which is so much information to just send with one little tiny laser beam, probably no bigger than the size of your fingernail. To date, we did not actually set up a quantum informatics setup just because we didn't have a lot of time. But this is going to continue in the future. Unfortunately, without me, I'm a little too busy in the fall to be part of that lab. But in regards to further study, I would definitely love to study optics in grad school. It is such a cool thing. And I really think that this experience prepared me to go into optics as a master's degree or even as a profession. I work in tutoring right now. I work tutoring particularly student athletes in math, and teaching in general is another thing that I'm really interested in. I find it to be really exciting. So to combine my knowledge of physics with the teaching would be really incredible. I want to particularly thank all of the funders for this research. Without you, I wouldn't be able to do this. I wouldn't be able to explore my love for physics. And I wanted to thank Dr. Darius Torchinsky as well for allowing me into his lab and allowing me to do all of the research that I did with him.
My name is Kayla. I'm a junior here at Temple University, and I'm also a computer science student. So the name of my research project was The Impacts of Identity on Ranked Choice Counting Methods, which revolves around this idea of ranked choice voting, or RCV. So this is an election method where we would be able to go to the polls, and instead of selecting one candidate that we think is fit, we'd be able to rank the candidates listed. So with this different type of voting method, there are a bunch of different counting methods and ways of finding a winner. So the most widely used in the United States is something called instant runoff or runoff. This goes by rounds. So first round, if no candidate has a majority, then the last place candidate is eliminated. We transfer the votes and then we move to a new round. And this continues on until some candidate has the majority. There's also another method called the border method, which runs by points. So let's say a first place vote is worth 10 points, a second place vote will be worth 9 points, etc. At the end of this, we would count all the points that these candidates have. Whoever has the most points at the end would then win. So with all these different counting methods, they tend to disagree with one another. Some methods will choose one candidate, another will choose another. So with this disagreement, how do we find the correct method to use to find a winner? So this is where our research comes in. So we wanted to know if there's any impact with social identity on these ranked choice counting methods. So we looked at race, gender, and political party. So the demographics of our study was primarily Democratic. This is because most of the elections that use RCV tend to be Democratic primaries. So we have an overwhelmingly amount of Democrats in our study compared to our gender split and our race split. So the most interesting part was when we looked at Oakland. Oakland 2010 was the first time they used RCV to find a mayor. And they had a female mayor for the first time, and since then they've only had female mayors, which then brought us to the point of gender and our connection with gender. So it seemed that in our study, gender had the most promising numbers. It looked like with RCV, male candidates were losing the RCV election since it was used, but they would win plurality. And plurality is our regular election method, the one that we're used to. And it was flipped for female candidates. So for female candidates, they were winning since RCV was used, but they were losing the plurality elections. So the importance of our research here is if RCV is to be used more widely across the United States especially, we have to know the real world impacts. And a good way of looking at the real world impacts is looking at social identity. So our next steps within this research, we would be looking at the order of elimination with these counting methods. We would want to see if there's any method that is eliminating a candidate first or leaving in certain candidates, and whether that has to do with gender or any other place they last on. So our conclusion so far that we've come to is that when these elections are close and there is disagreement, we have to look at the counting method used. The counting method used is important in that if there's disagreement or if the elections are close. And we also started looking at the fact that RCV makes non-viable candidates viable. There are candidates that wouldn't have really had a chance before, but with RCV being used, even if they're not winning, they're placing higher than they would have without it. So how have I developed? I have never done research before. This is my first time doing research. So through this program, I've been able to work closely with a mentor and feel comfortable working closely with the mentor and exchanging ideas. I've also learned more about Python programming, which is something I haven't done since I started programming a few years ago. And I've also learned LaTeX typesetting, which is a new skill I can take with me. So I would say thank you to the funders. Without your support, we wouldn't be able to continue to do research like this, and people like me who have never done research, this gives us an outlet to at least try and see if it's something that we want to do. And I also want to say thank you to my mentor, Jeremy Sivek, who's helped me this whole time throughout
Hi,
this.
my name is Nora, and I'm a senior physics major at Temple University. And this summer I had the opportunity to participate in the SURE program as an undergraduate researcher. And today I would like to tell you a little bit about that research. So in particular, I was working in the field of theoretical physics on a project titled Quantum Correlations of Quarks and Gluons within the Proton. It was based on the fact that for a while we've known that the proton is not a fundamental particle. It's composed of quarks and gluons, in particular 2 up quarks and 1 down quark. And we know that is true, but there's not a lot known about kinematically how they interact in order to actually form the structure of the proton. This is something experimentally that's being done at facilities in the United States and Europe, and also with the theory behind it in terms of actually trying to put together equations that represent the structure of the proton and how those quarks and gluons are interacting. So in my research, I worked with a mentor to actually try to confirm some of the constants in different functions that we call structure functions, and it was very tedious work aimed at hopefully kind of standardizing the equations we have that will experimentally be used in the future. I would say that what I learned tangibly in terms of skills, I learned how to document my work to look really pretty within LaTeX. I also learned how to cross-check my calculations in Mathematica. In addition, I learned how to dissect kind of daunting research papers, and I probably went through like 10 to 15 over the summer and kind of get out of them what I needed, and also check to make sure that my calculations were consistent with what other people in the field are finding. But I think that more so than any technical thing, I kind of learned how to deal with the frustrations that might come from research. My mentor told me that when you kind of feel frustrated about it, to think about the fact that you might be the only person in the world at the moment working on some very niche equation. And I found weird comfort in the fact that, yeah, maybe 1 day or 2 days or a week I made no progress, but at least I was focused on something that is impactful, and no matter how small it might seem, I think that it will be of use in the future. So lastly, I just want to thank my mentor, Andreas Metz, within the physics department, and his graduate student, Yorgo Sawaya, for their help in making a really intimidating topic feel more manageable for me, and also for helping me kind of figure out that I do want to apply to graduate school this fall and keep pursuing particle physics. And also I want to thank the supporters of the program for their funding and making this possible. So thank you.
Hello, my name is Kaden and I am a junior chemistry major. So the title of my research project this summer was Direct Synthesis of CF3 Carbonyls from Alpha Hydroxy Esters, Enabling Access to Fluorinated Bioisoteric Scaffolds. So the purpose of this project was to create a more effective way to add fluorines onto different molecules. By adding fluorines onto different molecules, you can kind of fine-tune a molecule's properties, such as its stability, its pH, its solubility in the body, and these are properties that are really important to medicinal chemists in order to develop drug molecules, because these molecules need to have very specific properties in order to do what it needs to do in the body. So my role in the project was to demonstrate that this technique was effective and that it was effective to a lot of different molecules. So I really spent the summer just doing the reaction and demonstrating it on a broad range of molecules. So through this I really got to learn a lot of different techniques, things such as doing reactions under inert atmosphere, doing reactions at really cold temperatures using dry ice. I got to learn how to purify my products using like column chromatography and recrystallization, and I also got to learn how to analyze my products using NMR, mass spec, and HPLC, all of things which are really important to research in chemistry. So my current plan is to stay with this lab and hone my synthetic skills. Through this, I'm really hoping that I can grow as an independent researcher and be able to start to make my own research questions. After I graduate, I'm planning to pursue a PhD in either organic chemistry or biological chemistry. But for the time being, I'm just excited to continue research in this lab. I wanted to end off by saying thank you to my PI, Dr. Daniel Kim, and my mentors for the summer, Ryan and Rifat, and of course also to the generous people who gave me this opportunity. Your support has been really instrumental in helping me develop as a scientist. Thank you.
Hi, my name is Amanda Lee. I'm a senior ecology, evolution, and biodiversity major, and this past summer I worked in Dr. Sewell's lab at the Temple Ambler Field Station. So my project is on looking at the soil seed bank and how that plays a role in forest regeneration. So with my project, so far some of the preliminary conclusions that I found is that a lot of the species that we're finding in the soil seed bank are ones that are from the surviving adults that have remained post-disturbance, and so things like tulip poplar, red maple, and a lot of the other woody species that were able to sustain the tornado are the ones that are contributing to the soil seed bank right now. So what this tells us about our forest is that ecosystems, especially old growth forests, can have a level of resilience to them, which is really interesting, and it helps us understand things like how climate change might affect these older ecosystems as we move forward. This experience and working in a lab has helped me prepare for graduate school. I'm hoping to pursue a PhD in ecology, and this was a great way to confirm that I like doing research and also gaining a lot of skills that will be valuable for the future. And not only am I learning technical skills and academically how to approach literature review and things like that, but I'm also able to build a support system at the field station. So a lot of them are helping me with professional development and working on my applications and just working on what plans I have post-grad as well. So this past summer I worked at the Temple Ambler Field Station under the guidance of Dr. Brent Sewell, and my project was looking at the soil seed bank and how it plays a role in forest regeneration. So I've been at the Temple Ambler Field Station since January of this past year, and since then I've been collecting soil samples and looking at how the species composition within the soil relates to the way that the forest has been recovering post-disturbance. So the Temple Ambler Field Station in 2021 was hit by a major tornado, and since then we've been looking at the way that the forest recovers and looking at different aspects like the seed rain, the recruitment species, and how other interactions have been affected since the tornado. This experience has been really valuable for me to gain fieldwork experience while also learning about how research is done in the first place, like how to design an experiment, and a lot of valuable skills that will help me in the future as well, like plant and seed ID. As of right now, I'm still working on processing the soil samples from this past summer, but I'm hoping that once I am finished with the data analysis for that, I can start working on comparing that to different aspects of forest regeneration that we're looking at. So things like sapling recruitment and the seed rain as well, and just exploring different dynamics that affect how how the seeds are stored in the soil. But yeah, this program has been really helpful for my professional and academic development, and I would like to thank the funders of this program, the CST SURE program for helping me with this project, and my advisor, Dr. Brent Sewell, for helping me develop this project and continue working on it.
Hi, my name is Jocelyn Colsey. I'm a senior biology major with a Spanish minor, and this summer I did a research project at the Ambler Field Station where I looked at if moss-forming biocrusts are able to facilitate the germination of a violet called the arrowleaf violet, which is the main host plant for the eastern regal fritillary butterfly. This project's important because this butterfly is very endangered. It only exists at one place in the whole world, called Fort Indiantown Gap, which is a military base. And its main host plant is the arrowleaf violet. So we're trying to figure out how to restore this host plant, which used to be found all across the United States, but now it's not found as much anymore due to habitat loss. And that goes for the butterfly as well. So the main finding of my project from this summer is basically that moss is significantly increasing the germination success of this violet, which is really good to know for future restoration efforts. In the near future, in my lab, we want to grow these violets outside because my project was in the greenhouse, and we want to see how the caterpillars of the butterfly are interacting with the violets and if they're able to use them successfully. And this research was a really great experience for me because I want to go to graduate school eventually, and I want to pursue ecology and get into like the policy and management side of things. So I feel like this opportunity to make my own research project and to work with a really endangered butterfly and help contribute to its conservation was a really good experience. I would like to thank my mentor, Dr. Sewell, as well as Chris LeClaire at Temple. And I also want to thank the summer undergraduate research experience for funding my project.
Hello, my name is Samar, and I'm a fourth year cellular and molecular neuroscience major here in the College of Science and Technology. And this summer I participated in CST's Summer Undergraduate Research Experience Program. And I had the pleasure of doing research under the mentorship of Dr. Shin Kong. And so the project that I did this summer was looking at the role of oligodendrocyte expressed ApoE in Alzheimer's disease. And so Alzheimer's disease, as you may know, is a neurodegenerative disease. And it's super important to study because it currently impacts about 7 million Americans. And that number is expected to increase as the population starts to age. And this is strongly influenced, the strongest genetic risk factor, I should say, for Alzheimer's disease is ApoE. And there's a specific variant, which is ApoE4, that confers the greatest risk. And so in the context of the brain, most of the research work has focused on a cell type called astrocytes, but there's another cell type called oligodendrocytes that has also been found to express ApoE in the brain. And this cell type is a cell type that is important for protecting neurons in the brain and making sure that the signal they transmit is transmitted effectively. And so that's why we're kind of focusing on this cell type. And so what we wanted to know in my research was whether ApoE that's expressed in these oligodendrocytes stays localized to the cell or whether it's trafficked outside of the cell to different cell types, and how that sort of influences Alzheimer's pathology. And so the way that we did this is we used these viral gene delivery systems in mice, and those systems expressed ApoE specifically in oligodendrocytes in our mouse models. And so the skills that I picked up sort of doing this research were I did surgeries on the mice to actually inject this vector into the mouse brain, and then I also did tissue sectioning, and then staining those tissues to look at them underneath the microscope to see what was sort of going on there. And so we have some preliminary data, And our preliminary findings are super exciting. They show actually what we sort of expected, which was ApoE is being expressed in oligodendrocytes and moves out to different cell types, mainly being neurons. So we do see that trafficking already. And so the work that I'm going to be doing this fall is to look at that expression more closely and to sort of quantify how much ApoE is being trafficked outside of the cell. So very, very good things to come. And so I just wanted to end off by thanking you for your generous donation and for the support you have given this program. It's so important to the work that my colleagues and I do, and in continuing to support the important research that we do. And so I just wanted to say thank you.
Hello, my name is Riley Keenan. I'm a senior here at Temple University, and my major is general science with education. And this summer I got the great opportunity to work with the SURE research program, and I was working with Dr. Mariana Bonfin, and her research looks at the tornado that devastated an old-growth forest on Temple Ambler campus in 2021. So my research was basically looking at the census data that we had collected years prior and putting this into a computer model that would predict how much carbon that the trees would sequester over a 30-year period. And then I looked at the relationship between the carbon sequestered in certain parts of the forest and maybe what species were able to sequester the most, and the relationship between species diversity and carbon sequestration. So that was really fun. The research involved censusing, which is like going out in the field, so the forest, and measuring trees and identifying species and such, which was super fun and an experience I never got to do before. So I learned a lot about identifying species, and certain parts of ecology that I had no experience in, which was really fun. So I'm really glad I got that opportunity. And I think in the future, working as a high school teacher, which is what I plan to do after my undergraduate experience, I think this is a really great opportunity for me because I don't think I'll be able to do research again. And I think this taught me a lot about using Python, coding. Yeah, I got to explore that more in depthly and learn some different types of like allometric equations and modeling. That was really interesting and fun. So thank you so much.
Hi, my name is Emily Gielach. I'm in my sophomore year at Temple, majoring in physics and chemistry. So my project this summer was the construction of a nonlinear optical scanning microscope. So the basic goal of this is to use a laser and other optical equipment to take a picture. The main component of this is the galvomere, which can oscillate very quickly in the x and y directions. So that moves the laser beam to different points on the sample and allows the image to be constructed. I learned so much this summer about physics and current research, but I also developed a lot of skills as well, like dealing with optical equipment and aligning lasers. But the most important thing I learned was programming. So I had no programming experience going into this summer, and that was definitely my biggest hurdle because I learned pretty much on the first day that programming is very important to experimental physics. So in the beginning of the summer, I spent a lot of time just getting comfortable with LabVIEW and MATLAB, and eventually I was able to develop the programs I needed for the microscope to function. I'm very grateful for the time I had this summer in the lab and just having the opportunity to make this project happen. So I'm going to continue to work in this lab during the school year and hopefully next summer too. And the next goal of the project is to start exploring acoustic wave propagation, which I'm very excited about. So I'm very grateful for this program. It has encouraged me to pursue a PhD in experimental physics after I finish my undergrad. So yes, thank you.
Hello, my name is Ijenu Okongwu. I am a senior math major at Temple University, and this summer I was working in a lab under Stephen McNeil as my mentor. The title of my project is A Map of Ice Raids in Pennsylvania. So the goal of my project was to create a resource for immigrant communities so that they could see where in their areas ice raids are happening, specifically in Pennsylvania. And an added benefit of this resource is that other researchers can use it to ask questions regarding the frequency or the location of ICE raids or demographic information of people that have been arrested at these raids. So starting in January 20th, 2025, since the increase in the quota for ICE arrests, there has been increasing fear in immigrant communities, and even more recently in LA with the protests and things like that happening, in addition to officers being able to stop and search people that they suspect may be in violation of any immigration laws. There has been an increase in fear. This is targeted at lessening the fear within immigrant communities, so they will be able to see where they might feel safe and where they might not. And for researchers, they'll be able to see the location as well as the kind of establishment where the ICE raid occurred. They'll be able to see the number of detainees that were apprehended at each raid. They'll be able to see if this information was available through either the ICE newsroom website or through any news report of the incident. They'll be able to see the demographic information of each person that was detained during the raid. And they'll be able to see the date. So if they wanted to answer questions having to do with the country of origin of most people that have been detained by ICE, they would be able to come to this resource and begin answering questions like that. So the skills that I have had practice with through this project have been map building. I've never worked with a map building project before, so this was a really good experience using the Folium and Pandas libraries through Python to build this map and put points on the map so that it could be a more interactive experience for people that are coming to it for further information. This was also a really good experience for design and getting practice with and asking questions, making sure that my solution was very targeted towards the issue that I was working to solve or address. So in the future, I'd like to continue working on this project. As of now, the interface for users to come on and put in their own experiences or their first-hand witnessing of any ICE raids is available, but we're not yet able to collect that information and use it to display new points on the map. And so far the only points that are available on the map are those that have been collected off of the ICE Newsroom website and from news reports. So upon graduation, I'd like to apply to both master's programs and industry jobs, and I'd say that this program has really opened my eyes to what being a math major can make available to me. So even though my focus is not on computer science and it's on pure math, I was still able to use the skills that I've learned to create a map using Python and the libraries that are available through there. And so I'd just like to thank my mentor, Stephen McNeil, and the organizers of this program, Professor Stefanili and Professor Varnum. And then in addition to that, I'd also like to thank the funders of this program for making this opportunity.